publications, very less of 1 or 2 publications, or even the study which has been done is either in 2 patients or 3 patients samples. So I had difficulty in to decide as to how to calculate or how to assess what sample size should be. Validation case is clear because where you ELISA, you have quantitative things and you can calculate with the whatever protein. If not with that disease, a similar disease where the mean and SC values are available, you calculate with the controls and the disease which to be clear. But discovery phase, is there any catch into it, or you just assume?
Sir, I would say there's no specific catch. They still follow the same principle that we discussed yesterday when you are doing the calculation of the sample based on the prevalence where we are able to identify, okay, the protein that you are localized is not due to a chance factor, just like we did the sample calculation yesterday. So same factors will come into play when we are doing the discovery phase also.
Thank you. Sir, maybe we can talk.
Yes. Thank you very much. Thank you very much for your patient listening, and please continue for the next sessions.
Thank you.
Thank you very much, and good afternoon to everyone. So this session is very small and very light, right? So let's proceed. In God we trust, right? So if you tell me there is God, I will not ask you to show me the data. But anything else you say, I will ask you to show me the data. Right? So any question that you have, whatever you have, anything you have, I will not, nobody will accept it till you show the data. Right? So we need data and we need to collect, right? So next. So when you can measure what you're speaking about and express it in numbers, you know what you're doing, right? So this is what he said. All science is about variables. Yesterday I said research deals with variability. No variability, no research, right? So all science, whatever, whether health science or social science or any science, it deals with variability, right? But you have to identify what variables. You need to identify what variables, keeping eye on your objective, right? And you need to measure it. You need to capture it. Those variables are nothing but attributes. So every variable has a value on each unit of measurement. And as a researcher, all you need to do is you need to capture that. If you have been able to capture that, what that value is of that unit of measurement, then that measurement is valid, right? And I told you yesterday, for any research question, at the end of your study, you provide answer. And for every answer, 2 questions are inevitable: how valid your result is, and how reproducible your result is. Your result cannot be valid if your measurements are not valid, right? For your results to be valid, your measurements have to be valid. And you have been listening internal validity, external validity, right? So how do you ensure internal validity? How do you ensure external validity? Internal validity you ensure by whatever you have got, is it actually there or not? External validity you ensure, can I extrapolate my finding to my target population or not? So that you ensure by doing appropriate sampling in case of observational studies, right? In case of experimental studies, there is no sampling. Where is sampling? So external validity is not an issue in experimental studies. It is an issue in observational studies. There, in experimental studies, randomization. Because those are not your random samples, they're samples of convenience. And if you are not randomized, you don't have right to use any of the statistical methods, which assume your sample to be random sample. That is why randomization is key, right? Of course, it has other benefit that I hope it was told yesterday. But anyway, so all measurements are not science. टेलर है वो भी टेलर also takes measurement, that is not science. Somebody वो भी measurement लेता है, ये क्या measurement, ये क्या measurement, but that is not science. But all science they deal with measurement. Next. Now look at this. We deal with variables, their attributes, right? So you have decided what my question is. You have decided that okay, for these questions I need these data, right? So look at this. Like Ramesh Kumar is a 37-year-old man. Now, this is, of course, this contains data, but you don't collect data like this. So in this, what are the variables? Name is variable. Everybody is not Ramesh Kumar, right? Somebody may be Suresh Kumar. Other is 37. Everybody is not 37 years old. Somebody may be 27 or whatever. Everybody is not male, right? Somebody, if you have one of your participants, it depends on your question, could be female. So there are three variables. So you collect data like this, right? You collect data like this, and you just don't go with a notepad and start collecting data. You collect data in a systematic manner, right? So what is that systematic manner? That is questionnaire, right? And how you should develop questionnaire, what should be the content of questionnaire. So those things we'll discuss very briefly, right? Now these are the variables we deal with. We have demographic variables, we have anthropometric variables, you have nutritional variables, genetic variables, what various domains of variables you have. And depending upon your question, you decide what domain to, what variables, and when to measure, how frequently to measure. You don't measure, see some variables you measure only once, some variable you measure repeatedly. Like gender, you don't measure during every visit. Age, you don't measure, right? Systolic blood pressure, you may measure repeatedly, isn't it? So those kinds of, you decide which variable to measure when, how frequently, and all those details you have to measure. Next. The bottom line is, we deal with only three kinds of variables, regardless of your question. Whatever your question is, right? Whether it is diagnostic study, biomarker study, or clinical trial, or case control, cohort, whatever is your question, whatever your study design, we deal only with three types of variables. And these three types of variables, you need to identify, and you have to put them on your questionnaire, and you have to capture for every individual, every unit of measurement. Your unit of measurement very often is an individual. In health research, is an individual, right? But unit of enrollment is individual. On them you are measuring values. But many times it's not like for an ophthalmologist. He enrolls 100 individuals and he says my sample size is 200 bilateral. 200. So unit of analysis is eye, isn't it? Most complicated is dental. So many teeth, and on each teeth, maybe on each quadrant, that's very complicated. Right. So you need to remember what is my unit of enrollment, what is my unit of analysis, right? So some data you may collect at individual level, like age. Even dentists, they don't calculate age for each teeth. Age is for an individual, gender is for an individual, right? So, but other things are at whatever level that you decide. So only thing. See, one thing I want you to remember, that just focus on three. If I read your proposal, and I'm looking at the variables, I will only look at these three categories of variables. What are these variables? How is he measuring? Right? So I look for outcome variable. I look for exposure of interest, and I look for other factors. Right? So if you're attending any research method workshop, then you need to know ABCD of that workshop. Like if you're learning English, you need to know ABCD. If you're learning music, you need to know Saregamapa. If you're attending research method, you need to know 5 things: outcome, exposure, other factors. These are the variables. And other 2 things are bias and roll-up chance, because you're dealing with sample. Every researcher is biased. Bias would be from beginning till end of your study. How do you frame a research question? From published literature, that itself is biased. If a study which has a conclusive positive kind of finding is more likely to be published. But it is not that all findings are positive. Some true findings could be negative as well. they have less chances of getting. So that itself, beginning itself is bias, right? And why are you doing research? You are doing research. You are spending so much time, so much energy, because you are convinced that yes, it works. Now let me show it to the world that yes, it works. Following rigor of research methods. So yourself is biased. Everybody is biased, right? And bias cannot be detected. So when I look at protocol, I look for biases. So there is no study which is free from bias. Best studies have minimum bias. And bias is throughout your research till publication bias, analysis bias. Everybody reports in the analysis section. I have done, they report the results of analysis, but they never say what all analysis they did, right? So they will do 100 analyses and whatever good comes, they take out. This is what I wanted to see. If there is a target, and if you take 20 stones and throw it, aankh band karke, 2-3 will definitely hit. And say, see, 3 times I hit the target with closed eye. How good I am, isn't it? So all those things are. So that is why in your protocol there has to be a section on analysis plan. In addition to data management, so in any protocol there are 2 sections related to statistics. One is sample size, and second is data management and analysis plan. What data you are collecting and how you are going to deal with the data that you have collected. All that you have to commit a priori, so that otherwise all you are doing is torturing data, and whatever good comes that you are reporting. Data dredging. So that is why they want you to commit a priori. That is why you must have seen in big trials, before you look at the data, there has to be data analysis plan. In the same hall, several years back, there was a huge trial of ICMR, and they were going to discuss the results. And Dr. M.K. Bhan was sitting here, late Dr. M.K. Bhan, and he said, "Where is the statistical analysis plan?" They said, "No, we don't have." Then he said, "Stop the meeting." In 10 minutes time, we stopped the meeting, and he said, "No, you develop a statistical plan and go and get it approved from data system or DSMB, and then you look at the data, right? Because they don't want you to, everything they want you to specify a priori, right? So those things are there. So 3 variables: outcome, exposure, and other factors, and you need to capture them. So next please. Variables, every variable, see, these variables, they have their own position in the framework, right? So I just take an example. Suppose I'm interested in knowing the role or relationship between smoking and hypertension, right? So then there is a position. The smoking leads to hypertension, right? So smoking is exposure, hypertension is outcome. It is not other way that person becomes hypertensive when he picks up smoking, isn't it? So it's not that way. So there is a direction, but smoking is not the only factor. Any outcome you take, whether communicable diseases or non-communicable diseases, there is no single outcome. COVID, there is no single outcome, right? Any outcome you take, there is no single outcome. If there is no single exposure, single factor, and we wish there was single factor, we could have bargained well with that factor and dealt with nicely, right? Most of the factors we do not know. We know very little about the factors. That is why every day, we keep discovering new biomarker and new factors and all those novel, all that we keep discovering, right? So we know very little, and even very little what we know, we cannot collect data on all of them, right? We cannot collect data on all of them. We only collect data on few of them. So only 3 variables: outcome variable, exposure of interest, and other factors. Your other factors can be confounders, isn't it? They're all nuisance. And you have to spend more time on dealing with nuisance. I'm not interested in knowing. See, your most questions are bivariate. What is the role of this on this? What is the role of this on this? But there will be a whole lot of nuisance. They will not let you know the true relationship. They will confound, they will distort. So you have really spent maximum of your energy on these nuisance. So you need to identify them. And once you've identified, then you need to deal with them analytically. Or maybe you deal with them at the design stage. So there are set rules for that, right? So there are confounders, other factors. there are effect modifiers, there are intermediate variables. So intermediate variables are nothing but your outcome variable, process indicators, right? Say suppose, for example, I am interested in role of, let's say, fat, saturated fat on, let's say, MI, right? So in between, there will be a whole lot of factors. Saturated fat will lead to BMI, saturated fat will disturb lipid, and they are predictors of MI. So in between, they are intermediate variables, they are process indicators, right? So process indicators, you don't, if your question is this versus outcome, then intermediate ones, you ignore them. They are not confounders. If you adjust for them, then the analysis that you are doing is like Kalidas analysis. Like you are cutting the same branch on which you are sitting. You will get your results will be towards null. Right. So you need to identify what these variables are. Right. It is not that every third variable you need to adjust. So these are the three category of variables, and you need to collect them. Every study will have outcome variable. Even diagnostic test study, isn't it? Whether it is prevalence study, there is outcome variable. Incidence, outcome variable. Every study will have outcome variable. Outcome variable is in which you are actually interested in. You cannot say my study has no outcome variable. So that means I'm not interested. And why are you doing the study? Every study will have at least one outcome variable. Invariably more than one, right? Then exposure. So these are one, and on them you collect data. So data basically always collect data on these variables. When you capture data, always capture at its original level, not at modified. Say, for example, age, you can, for gender, you will collect data either male or female, right? Isn't it? You have no other option, right? But quantitative variables, you have option. Like, for example, age, you can collect data in decimals, right? For 24.63 or whatever, right? In years and months, you can collect. You have option of collecting data in completed years. You have option of collecting data in interval 10 to 20 years, 21 to 30 years, 31 to whatever, isn't it? So never collect data on quantitative variable in categorical form. Always collect data in original value. Grouping you can do later. Because if you collect data in of a category quantitative variable in categorical form, then later, suppose age 10 to 20, you will never know how many were 10 to 15. But if you collect in quantitative, you can always create 10 to 15, 10 to 17, whatever you want, right? So always, so that was the message. So outcome variable, always quantitative. Exposure variable, collect in quantitative if it is quantitative. It can be categorical also. Your outcome variable can be categorical, yes or no. Your outcome variable can be quantitative, birth rate of newborn, systolic blood pressure, whatever, right? So outcome variable can be categorical, can be quantitative, can be time to event. I told you yesterday. Your exposure variable can be categorical or can be quantitative, right? Your other factors, some of them can be categorical or some of them can be quantitative. So when you capture your data, because there is no second opportunity for you to recapture the data, except that you have some stored sample and you redo it and those kinds of things. Otherwise, from collecting data from participant, you have no second opportunity, right? And even if you have second opportunity, that's not the data. That by it could have got modified if it is that kind of variable, right? So collect your data at its original form. That is what this slide I was going to tell you. So three variables: outcome variable, exposure of interest, and other factors. And then how are you going to collect? That you will explain in the method section. And collect at its original form, at its original level. Next, please. Next. Let us skip this. Next. Okay. Now I told you your measurements have to be valid and reproducible. If not so, then your results cannot be. No statistical method or sophisticated technique can make your results more valid and reliable. Your data is not valid and reliable. Right? If your data is valid and reliable, and if I do wrong analysis, somebody else can do right analysis and the results will be valid and reliable. Next, please. So type of questionnaires. In your questionnaire, you have different kinds of questions. I have not seen any study, maybe in the last 30 years. I have not seen any study which has collected whatever data they have collected, they have actually analyzed all the data. And whatever data they have actually analyzed, all of them they have reported. Then why do you collect data? Nothing comes free. Nothing comes free. You have to pay price for it. Isn't it? When you're collecting your data, don't collect what you can collect. Isn't it? You should collect data on keeping objective in mind. So there is a basic rule for collecting data. People think that सबसे पहले questionnaire develop कर लो, right? Let me develop the questionnaire. There is a golden rule for developing questionnaire. Golden rule is you know your objective. You know your objective. Please prepare your dummy tables. Think of all dummy tables which will go in your report and you can generate, keeping the objective in mind. Dummy tables are final tables minus the numbers in it, right? So to fill these tables, whatever questions you require, only those questions should be on your questionnaire, right? Only those questions should be on questionnaire. So if any question that is not participating or not required for any of the table, that is not, better not to collect data on that. So do that kind of thing. So questionnaire will be, see, in questionnaire, you have two kinds of things. One is initial part, it is not meant for analysis. Those are identifiers. Like, isn't it? Those are identifiers. What is your name? What is your mobile number? What is your ad? Those are identifiers not meant for analysis. Right? Now, and then after that, you have questions which are meant for analysis. And these questions are nothing but variables. Age is one, that's variable. Gender is one, other variable. Right? Height, weight, this, that, all that. So these are basically your variables. Now for each of the question, see there are different ways of capturing question. Easiest way, so there are some variables which are very easy to measure. And there are some variables very difficult to measure. Right? Easy to measure is gender. Like if you're doing a study, you don't ask a person, are you male or female? Look at the person and right, isn't it? Right? Age, you have to ask that person what is your age. Right? Again, it depends from whom you're asking, because your aim is to capture that valid measurement. So if you ask my age, so whatever I say, you'll record it. Right? But if you're asking age from, you're doing study, community-based study, woman, uneducated, if you ask her age, she has no idea about age. So you may have to probe and ask multiple questions. Isn't it? How many children you have? What is the eldest child? So you have to ask multiple questions so that at least you reach closer to her age, true age. So again, it depends the context where you're measuring, right? So if simplest, then some like quality of life and construct those kinds of latent variables, very difficult to measure, right? So there's no single question which can measure. So that is why you have tool to measure them, right? That is why you have tool to measure them. Now there is a difference between a tool and a questionnaire. There is similarity between the two, and also there is a difference in the two. Both tool and questionnaire, they contain set of questions, right? They contain set of questions. In questionnaire, you analyze question by question, but in tool, you analyze not the question by question, but domains, like say quality of life. You may have 5 domains, each domain contains whatever number of items, which you may capture on Likert scale or whatever scale, isn't it? And then you analyze total. That is what is tool. So tool is different from questionnaire, right? And in any study, whether you are doing in using a questionnaire or tool, it has to be tested, it has to be validated. So we'll come to that. Next. So questions will be different kind. Like one question, like quantitative variable, you don't code it, capture it like this. Say for example, age, and you know age, you can have 2 boxes. If you want to capture in completed years, or 3 boxes if you think more than 100, right? Systolic blood pressure, you will have boxes. But for gender, you know what are the possible responses because you want to minimize your time. Now you have options. Gender, either you leave it as blank and you write male, female, it takes more time. But if you know that gender possible answers are male, female, you write it ahead of time in the question itself so that just tick. Just take, you save your time. But some questions, you know that answer for this, you don't know complete answer. But you know that almost 90% of respondents will give one of these four options. So you quote them, but you keep an option of 5 that others is specified, right? So that is basically mixed kind of thing. So that finally, once you have finished your data collection, then you visit this object option and see what are the possible answer. So you are collecting this in qualitative manner. Fifth one is qualitative manner, qualitative, and you analyze this the way you analyze the qualitative variable. In this, you will write in words. People use different words to convey the same thing. So that's why you combine and create a domain kind of thing. So eventually, with 4 categories, now you may end up with 7 categories, right? So that is how you do that. But if you know that these are the possible options, you basically do that. Every questionnaire should be valid, reliable, all that. Every questionnaire should have a unique ID number. Every questionnaire should have a unique ID number, right? And unique ID number should be written on every page. Because if you're doing a study for 1 year, 2 years, वो page फट के कहीं चला जाएगा, right? So if you find some loose page, at least you should know to which ID it belongs to, right? So that you have an opportunity to stitch that. So write unique ID number on every page. It doesn't take much time, right? Then when you fill your next, please. Let me finish. Next. Yeah. See your flow of questions. When you prepare your questionnaire, your starting question should not be your main question. There is some warm-up question type, isn't it? Warm-up question, so that your main question somewhere in the middle. Main question should be somewhere in the middle. No questionnaire should be very long. Longer the questionnaire, people will say bhar lo, isn't it? So your quality of data will be affected, right? And also your questionnaire should be in a particular sequence. Your questionnaire should be context specific. Your questionnaire should not be like how many cigarettes do you smoke? And then next question is, are you a smoker? Right? It should be, oh, bolega, what is this? Right? Your questionnaire should not be how many children you have, and then you ask, are you married? No, this kind of question you can have if you're doing study in US, but not in India. So it should be context specific, right? And in every questionnaire, you should have all that jump kind of thing, whatever is not applicable. So if person doesn't have this problem and next 5 questions are linked to those who have this problem, then there should be skip option, and all that should be clearly explained in your question. So let me go to this. So basically, there's a brief. I'll give this slide. Brief principle. These are different kinds of questions. Also, see, some questions would be single answer and some would be multiple answers, right? Single answer would be, say for example, one question is, from where did you come to know about this workshop? So multiple responses, isn't it? You came to know from this source, you came to know from that source. So for this question, there could be possible answers. So you may take more than one. Right? So those are multiple kind of things, right? But this, when you enter data for this, each option has to be a separate column, separate variable. Think of Excel sheet. So suppose I'm collecting data on non-communicable diseases, right? So somebody may be hypertensive, diabetic, stroke we have. So all 3 I cannot write in one question, isn't it? So hypertension has to be one column, diabetes has to be another column, isn't it? And then later at the time of analysis, I can see who all have how many multiple comorbidities or those kinds of things. But collect data, when you capture data like this, systolic blood pressure, diastolic blood pressure, on your card, you write 120 by 80, 120/80. But when you enter your data, it has to be 2 different fields, 2 different columns. 120 is one, 80 is other one. Right, next please. And then see, some of your questions can be captured as yes, no, some question can be captured as quantitative, some questions can be captured as mixture of this, some categorical and then open-ended. Some question can be captured. So tools basically, tools can be on your likelihood scale. Isn't it? You are familiar with this, right? So tool is something different. Tool is something completely different as compared to questionnaire. Tool is, if you want to develop a tool, that itself is a standalone research. To develop a tool, you have to go through different steps of developing tool, isn't it? Face validity, content validity, construct validity, all those kinds of things, reliability. So those things you have to, that's a different kind of line. But when you develop a questionnaire, it is always a good idea to pre-test it in the field, so that if there is any kind of, there could be some problem in construction of sentences, maybe person is not able to understand what you're asking, isn't it? So always good idea to pre-test it. When you pre-test it, you may realize that some important question I have missed. I need to add that also, right? Or maybe you may realize, no, this question and nobody is answering, or this is not important. So you may drop also. So you can modify all that, isn't it? So it is always good idea to do a pre-testing of the questionnaire. There are some broad rules for this. Questionnaire, as I said, it contains questions, some for analysis, some not for analysis. Decide to include essential questions. That means you make your dummy table first, and then work backwards. To fill those dummy tables, whatever questions are required, only those questions should be on your table, on your questionnaire. And each subject should have a unique study ID, right? Also, see, when you're doing a big study, then you may have multiple questionnaires. One questionnaire for demographic, one questionnaire for clinical, one questionnaire for nutrition, one questionnaire for this, one questionnaire for that. You can have multiple questionnaires, right? There has to be common unique ID number on all of these, so that later you're able to stitch all the questionnaire for a subject together. So there has to be common field, unique ID on every questionnaire, right? So that you have to have also even so clinical variable. If you're doing follow-up study, then maybe 5 visits, and for each visit you have one schedule, isn't it? So then all these 5 have to be stitched together along with other performance. So unique ID has to be there on every performer. On every schedule, you have to have unique ID number, right? Order of questions should be in sequence. Variables should be recorded at highest level of measurement that we have already discussed. Derived variables should not be in the performer. BMI. Derived variables are those which you're not measuring, you're creating. Like BMI, you're not measuring. You're creating by using some 2 variables in some way. Height, weight, and weight by height square, right? Hypertension. Hypertension is not a measured way. You don't measure hypertension. You derive hypertension, right? Using what is systolic blood pressure, what is diastolic blood pressure, is the person is an anti-hypertensive drug. So look at these traces together, and then you say hypertensive or non-hypertensive, right? So don't have derived variables on the questionnaire, right? So BMI, if you have a questionnaire, while calculating, you'll make some error, isn't it? While calculating, you may make some error, or whatever BMI you calculated, if you enter, you'll make some error, right? So there's no point. So this you can always do it in your finally, just give one command and BMI will be calculated. So never have direct variable on your questionnaire. Now, if more than one group, same proforma, so you have to have same proforma regardless of the number of groups you have. Only difference would be that you will have a variable which will indicate group A or group B or group C, right? Even there could be some set of questions which are not applicable to one group, like you have one group as disease, other group as non-disease, and you are collecting data information related to disease, it will not be applicable to non-disease. But still you have to have same questionnaire, right? And because you're going to stitch the 2 together. For analysis, only one sheet has to be there. In your study, you may have 10 sheets, 10 questionnaires, but when I analyze, I will put everything into one. Because analysis has to be done together, not separately. Some analysis could be separate, but I have to look at everything together. So I'll stitch everything together, right? So It cannot happen कि वो systolic blood pressure cases का उधर है और उसके नीचे diastolic आ रहा है. This variable has to be same, right? So even if it is not applicable, leave that blank. Pre-testing of pro forma is mandatory. If you are using questionnaire developed elsewhere, pre-test it. Pre-test it, right? You may do most of the time. People come to me and say, "Sir, this is my data. So can you analyze?" I say, "Okay." And sir, this is my mother article. This is my mother article and this is my data. So that means you are just replicating and उसी का questionnaire use कर लिया. This questionnaire may be in different setting and some questions may not be relevant in your setting, or maybe some questions are relevant in your setting, which are not in existing questionnaire. So all those things you need to definitely translate, back translate. If you are doing multi-centric studies, right? Like to start doing in Tamil Nadu, Kerala, and these and other states, language is not the same. So same, you may develop questionnaire in English, but it has to be translated if you're doing community based studies, right? So then you have to back translate it just to ensure that in the process of translation, you have not missed things, isn't it? Or meaning has not changed. So this thing has to be done independently. Translation and back translation. Not that she translates and she back translates. No. If she translates, somebody else should back translate. And then see, are you getting back to same questionnaire or not? Right? So it has to be done independently. Then questions can be pre-coded, all that is fine. Multiple answer. Finally, a good pro forma, please consult somebody who is going to do your analysis. Because it should be analysis friendly. It should be, because it has to be analyzed. It has to be used for analysis. So do consult somebody or pay attention from the analysis point of view. So data collection, I think I will, all that is fine. Next. I will just spend next. I'll just go to one slide. Next. Now, okay. Now see, most of you, you would have so far used Excel sheet because the kind of data that the volume of data was not heavy, right? So if the volume of data is not heavy, then you can use Excel sheet. But Excel sheet has its own advantages and disadvantages. Advantage is there is no learning time. You can get started immediately, right? But disadvantage is that there are errors which you cannot control for. There are some errors which you cannot control for. Excel, anybody can enter. In 5 minutes, if you explain to somebody, but there are some basic rules if you're entering data in Excel. I have seen even for big studies, people enter their data in Excel, right? But I would discourage them using data in Excel for big studies. So there is other option. I will tell you that. But if your study is small and you're entering data in Excel, so there are some rules. See, analysis does not take any time. Analysis takes no time, but more time takes in preparing your data file. Right? So if you have not entered your data well, right, I will not do the analysis because it takes more time to prepare your data. So why not enter your data which can be analyzed easily? So Excel sheet, rule of thumb, your field, you can use only row number 1 for name of the variable. And name of the variable are in column and it is unique. It is unique. Name of the variable cannot start from number. It has to start from alphabet. So in your study, if you have 100 participants, so in Excel sheet, how many rows you are using? 101, isn't it? If you have measured systolic blood pressure 3 times, first 1 month, 2nd month, and 3rd month, then how will you name your variable? You cannot say 1 SBP or 2 SBP or 3 SBP. You have to say SBP1. Your variable can be alphanumeric, but not numeric alpha. So SBP1, SBP2, SBP. You don't need to write systolic blood pressure. Everybody knows what is systolic blood pressure, right? If you write SBP, you know what it is, right? And it is very bad practice. It is very bad practice to enter your data in coded form and write all these codes in a sheet and keep it at some place, very safe place. And when you need this, you don't get it. The most important thing in our house, we don't get it when we look for it. Because we keep it at such secret place, you don't know where it is. Right? So it is not good to enter data in coded form. Right? Some, like say from the gender, you can write MF because we know this is mild or this is male, female, all that we know, right? But some variables where we don't know, it's not common sense. So and it's better to enter your data as such, right? Like severity of disease, mild, moderate, severe. Mild could be M, moderate could be M, or severe could S. So you need to differentiate. You need to differentiate. So, and also see, in this, what are you entering? You are entering quantitative variable, categorical variable. You cannot, it has to be either alphabet or numbers. It cannot be both. Like, for example, age. So just click this two times. Next. Okay, so the above one is right way of entering, below is wrong way of entering. So below, see, for example, age 40 years. You can't enter like this because number and alphabet not allowed, right? So you have to write age as 40. But you are saying no, I want to be after 10 years. I want to know age was in years, not in months. So then write age YRS, right? Age so that you know what it is in years, right? Then, say for example, sex male M and other one is male, other one is small m. So if you decide to enter in, write in quotes and write capital M and a small m, then the system will take these as two kinds of males, bigger male and smaller male, right? So don't do that. So you can write MF, either lowercase or upper. Stick to one case, lowercase or uppercase. Then, no matter what data you have, there will be missing value. There will be missing value for whatever reason. Isn't it? For whatever reason, either person did not respond, or maybe the sample you collected was inadequate, or sample got spoiled, or whatever happened, isn't it? Or maybe person did not come for the follow-up, right? Or person left the study, or whatever happened. There would be missing value. Definitely there would be missing value, and you need to clean your data. You need to clean your data. There is no data which is completely clean, right? So whether it's like rice or gehu or whatever, we need to clean it. We need to clean it. You can't bring from the field and say this is clean. You have to clean it, right? And just imagine best quality rice, if it is not cleaned, and if you cook, ek do stone aage kuch aa gaya, see, bad, right? So collecting data is a very painful exercise, very laborious exercise, right? So you have to clean your data. If you have not cleaned your data and you say it is clean, that means you have not looked for it, right? You have to look for inconsistent. There will be error. Absolute ideal data is not the real data. That is simulated data, right? So spend some time. Analysis does not take much time. Spend your time on cleaning and preparing your data. If you have done that well, then later part is fine. So those are basically. But if you're dealing with large, I think this is last. Next, please. Okay, you can enter your data. You can enter your data anywhere, regardless of which statistical software you're going to use for analysis. Enter your data wherever you feel like, right? So if your study is small, you can enter it on a spreadsheet, but the spreadsheet has limitations. You cannot have relational checks. You cannot put relational checks, right? But if you enter your data in other systems, like say, for example, APInfo or MS Access, right? So there you can put relational checks. You can put range checks. You can put jumps, right? You can put those several checks you can put in. Like suppose you know that gender, your possible values are M and F. That's all. So anything other than this, if you put, it will stop you. It will not allow you to enter. But if same thing if you do in Excel, you can put A to Z anything right. It will not stop, right? Range checks. If you know triglyceride cannot be more than this. It was 150, but by mistake you type 0 2 times, so 1500. It will stop you. In Excel, it will not stop you, right? And some of the not applicable fields you can enter in Excel, but in those it will not allow you. So it is always better to use for larger studies. For larger studies, You develop, those are called data entry templates. Data entry templates means you develop your questionnaire on paper. Suppose it has 4 pages. So you'll have 4 screens. Those screens are nothing but photocopy of your questionnaire, right? And just go on entering tabs, like as if you're entering in a questionnaire. But checks will be there, built-in checks will be. So that is always better. Now there are 2 methods of data collection. One is you collect on paper and then you get it entered through some data entry operator. And other is on tab itself. You are capturing data on tab itself. But there are limitations for this. Once you collect data on tab, you have no way to verify. Go back to the source. This becomes your source document, source kind of thing. But once you collect data on paper, and when you enter it, there could be entry error. But that entry error you can go back to your source documents and verify what is right, what is not right. Right? But again, on question, if you're collecting data on paper, you have no idea that when the data was collected, did he go to the household or not? या पेड़ के नीचे बैठ के भर दिया उसने या पेड़ के नीचे बैठ के सारे फॉर्म भर दिए उसने. But when you collect data on tab, location is there. Location is there, and you know that at which location, at what time, right? This was filled. So all those checks are there, right? So this is the advantage. Tab, when you enter your data on tab, you may have 5 different centers. Each of the centers are collecting data on tab, and all that data is coming to ICMR immediately, right? The moment there is a connectivity, it is immediately transferred to wherever is the data management center. Big studies always have a data management center. Big studies, multicentric studies always have data management center. All multicentric studies, your questionnaire has to be exactly same, because this is one study. 5 centers cannot have 5 questionnaires. 5 centers cannot have 5 questionnaires. It has to be exactly same. But in some situations, they may say that, okay, at my center, I will do this core part, but let me add another 5 other questions which are specific to my center, so that depending on the funding agency, that may be allowed. But core part is exactly same in all multi-centric studies. So I know it's getting late, so I'll stop here. So if there is question, we can take.
This one question is coming, like how to improve the data quality when we are conducting a multi-centric study, sir?
Yeah, see, basically, if you're doing a multi-centric study, all multi-centric studies, by rule, they say the PI of the site should, on the same day, should sign on every form that yes, he has checked. No one signs. Most of them, they don't sign. They don't have time. And many of them forget about signing. They get a stamp made of their signature. Or sham ko har ek stamp laga diya. That is not it is, isn't it? So you have to, the PI has to actually check, isn't it? Also, when you are collecting data from the field, whoever is collecting, there is a supervisor kind of person. So he ensures what you are doing, right? So that is how basically you train. Otherwise, all studies, you have to train them well, isn't it? It is not that any field worker or any person can go and collect data. They have to be trained, and you have to make sure that now the training has to be evaluated, right? So, and also, if you're doing multi-centric study, the clinical trials, you also should develop kind of system of follow-up, a reminder kind of thing. Kiska reminder kab, kiska follow-up kab due hai and all that. Because you don't want to have missed visits, right? So each study should have kind of pop-up kind of thing. हाँ, इसका follow-up due है, इसमें तो उसको remind भी करना है, so that he or she comes. So those are the kind of things. But multi-centric studies are not as simple to do as that, right? As far as biological samples are concerned, anyway, you'll follow the same rule. You may have external quality assurance and all those kinds of things, right? But also one more thing. So that is one. Many of the like biochemical parameters, many of these biomarkers or whatever you call, see, to measure them, you have some kits, and every kit has a minimum detectable limit. Every kit has. So what do you do there? if whatever you're measuring, that is either, so there can be two possibilities. It's there or it is not there. And second is, if it is there, then it has to be captured. If it is there, it could be 0.1, 0.2, 0.3, whatever, up to whatever. And to capture that, you have a tool, you have a kit, and that kit has minimum detectable limit, right? And minimum detectable limit is, let's say 5, for example, right? And you have 100 participants. And for 10 participants, it is below that minimum detectable limit. Then what do you do for them? Do you leave them as blank? If you leave them as blank, you're losing on numbers. They have some numbers below 5. They have some number below 5, which your kit is unable to detect. Right? It is not so sensitive to go that below down. So the rule of thumb, so there are two options. One is you leave blank for them. Second option is, no, there is some value. There is some value which your kit has been unable to detect. Safest thing is assign that middle value. I will assign 2.5. I will assign 2.5 for everyone. Right? If it was possible to actually capture all possible values, then maybe to someone where I was assigned 2.5, it was 1 or maybe 4 or maybe 3. So, but on an average. So analysis is all, because final analysis I'm going to do is average, right? So it will average out. It will average out, but I will not lose on numbers. So that is what you have to tell that person that, look, some value is there, but my kit is unable to go below that. So you basically interpolate the value. You interpolate the value and mention so that you don't lose on numbers.
Sometimes when we collect data, like we have example, systolic blood pressure, we get some outliers, like they will say 240 or 300 or something. So how to deal with these outliers?
Yeah, see, again, outliers, once if it is some value which is away from the group number, isn't it? So then there are only two possibilities. One is there is a keyboard error. There is a keyboard error. And second is, yes, I think you have to look at his other parameters, other conditions, isn't it? And that condition may indicate, what is serious patient hai, ye hai, wo hai. So it is possible to have that kind of value, isn't it? It is possible to have some kind of value, but you cannot have 200 or 2000 as a triglyceride, isn't it? वो 200 का फिर वो लगता 0 दब गया, right? So 2000 तो नहीं होता है, but there can be 400 triglyceride or 300, but that should other variables should support that, isn't it? So you need to check other variables so that you get convinced that yes, it's possible. So all you have to do is that value plausible or not for this particular patient, right? Yeah.
So can we leave these outliers or we have to include it as
we
are not sure?
No, see, all you have to do is you have to ensure that this value is plausible. Right? If there is no keyboard error, right? And if you looked at other parameters and if you think कि भाई ये हो भी सकता